Okay, welcome to lab. Since you can't come to lab, lab is coming to you. So we are on lab 4, and this week we are going to quantify the phosphate from our lipid purification that we did last time. So what I'm going to do is I'm going to give you the normal lab spiel, and then I'll actually walk you through the experiment. So you'll have 2 parts to do. One is just the lecture, and then the other will be the lab. So when you're writing up your lab reports and stuff, you can call what we did last week day 1, and then what we're going to do today is day 2. So you need a couple of different reagents to do this assay. You'll need your concentrated perchloric acid. Remember, that's what you added to your samples last week. You'll need ammonium molybdate, and it's important that you make your ammonium molybdate at 25% weight by volume. Then you're going to need ascorbic acid, 10% weight by volume. And then we need a way to quantify how much phosphate we have. So remember last time, not last time, time before that, when we did the spectrophotometer and we were looking at absorbance of different proteins, when we had BSA and we didn't have an extinction coefficient for BSA, we had to do an indirect method where we created a standard curve and then we're able to identify our unknown. We're going to do something extremely similar to that today, and we'll have a sodium phosphate standard. And so that sodium phosphate standard is already made for you, and it is at 1 microgram of phosphate per 100 microliters. So your first step in the lab is to make your reagent solutions. The perchloric acid is already at 70%. That was what was in the hood. Ammonium molybdate, 25% weight by volume. Weight by volume is always going to be in units of grams per milliliter. So you would put 2.5 grams of the ammonium molybdate per 100 mils of DI water. And to make this go into solution, you're probably going to have to heat it and even possibly filter it. But it's already done for you because you're not in lab today. Oh, so nice. All right, so ascorbic acid, 10% weight by volume, that means 10 grams per 100 milliliters. You'd make that one up. And then the standard is already made for you. So the first point of the lab is to make our standard curve. We want to be able to convert absorbance at 280 nanometers, that should be nanometers, to concentration of our phosphate. So when you make your standard curve samples, you're going to use these tiny disposable culture tubes. And we're going to have an incubation period, and so we're going to have to put them into a beaker of hot water. And so if you ended up labeling these with tape, the tape would come right off. And so you would just write it. You can't really see that, but you write it on the tube with a Sharpie. And then when you're done with it, all your samples go down the drain, and then your test tubes go in the disposable, well, in the glass, broken glass container, right? Okay, so here's our table. This is the first table that you're going to create for the lab. So we need to vary our concentration of phosphate inside of our standard. So of course our first one is going to be 0. So when you're making these standards, we want a final volume of 3.3 milliliters inside of each tube. So whatever you add in terms of microliters of your standard, you want to make the total up to 3.3 milliliters. So you're going to fill it to 3.3 with DI water. So if your standard is 0, you're going to put 3.3 mils of water. A standard of 1 is going to be 100 microliters. If you want 1 microgram of your sample, you put 100 microliters of your sample, which means you would only need 3.2 milliliters of water. And so on and so on. You're going to go all the way to 5 micrograms in your last standard. Now, if you remember from last week, when you made your TLC samples, you took each of these, you scraped them, you put them into a tube, and what did you do? You added 0.65 milliliters of perchloric acid. You did this last week, right? So we want our standards to have the same thing that was in our samples. And so what you're going to do is you're then going to add 0.65 milliliters of that same 70% perchloric acid to your samples. And then we want to create a color difference that we could monitor through spectrophotometer. And so we're going to add 0.5 milliliters of 10% ascorbic acid. And then we're going to add another 0.5 milliliters of 2.5 ammonium molybdate to each of our samples. And so we will go and take those and read the absorbance of those values, and this is what will create our standard curve. We'll graph absorbance at 280 versus micrograms of phosphate. So while you're making your standards, you also need to prep your TLC samples to be read. So you've already added, this is your scraping, right? You added your scraping, you added your perchloric acid, and that's why I have those grayed out. Then you're gonna have to add 3.3 milliliters of water to every single one of your samples. And then you're gonna add that same 0.5 milliliters of ascorbic acid and the same 0.5 milliliters of ammonium molybdate, so that all of your samples are prepared exactly the same way. And then you would go in and monitor absorbance at 280, okay? So once you've added all of your reagents to your samples, you need to incubate them in a hot water bath. Ooh, you need to vortex and then vortex. Then, forgot about that. Place all your samples into a hot water bath and let it go for about 5 minutes. It should be on a setting of 5, and you can see that hot water plate, that hot plate's going in the background. Then you need to make sure that those samples cool before you go and try and read them in the spec, because you can create condensation on the side of the tubes and it can alter your results. So you allow them to cool to room temperature. If you just put them in a water bath, they'll cool down relatively quickly. And then you transfer your samples to your cuvette, read your absorbance at 280 nanometers. Now, you want to blank on your 0 standard. It's important that you keep that blank for both your standard samples and for your TLC samples. So if you were reading it on the screen, and I'll show you, I'll take you and do it, don't clear the screen, keep your 0. Then you're gonna create your figures. So you're first gonna graph, excuse me, your standard curve data, and that's absorbance at 280 versus your micrograms of phosphate. This is just a pretend graph that I made. It's not right. It's just to show you what I'm looking for. And then you have your data points. You plot a linear regression line between your data points. You get your equation of your line, y is equal to mx plus b. And from using that equation, you're able to solve for your micrograms of phosphate in your unknowns. And so here's your table that you're going to put. You'll put your absorbance value of your unknowns, and then this is the calculated micrograms of phosphate that you used in this equation, that you derived from this equation, right? Now, it's hard to understand micrograms of phosphate. That doesn't tell us a whole lot. What we really want to know is what percent of all the phospholipids, right, inside of bovine cardiac tissue were each of the individual phospholipids. So what you do is you total all of these. You take everything that was in this column, you get a total, you get a sum, right? So to find your percent, you take your micrograms of your individual, so in this case it's DPG, divided by your total, and then multiply by 100, and you get your percent of your total. And so what I'm going to want you to graph is this percent. So you're going to make one more graph for me. You're going to graph your percent of each TLC unknown in the bovine heart sample. And it will just be a bar graph that sort of looks like this. And you'll have some columns that are big and some columns that are not so big. But I want you to see the distribution of the different phospholipids within the sample. So it's important when you do this to understand how this relates to published data that's out there. So what I want you to do is I want you to go out on the web, find a good verified source that has percentage of our different phospholipids in membranes from bovine cardiac tissue. And so your lab report in the end is going to be one big lab report from last week. So you'll have all of your day 1 where we did our purification and separation. And then you'll have today, day 2, where you have your quantification. And then you're going to create one general conclusion after all of these tables and graphs. And don't forget to include that literature search for those published values. And that's it. Okay, I'm going to start the experiment. Okay, so here's our experiment. I have my 5 samples here that will have my phosphate standard, and then I'm going to have my blank. All I've put in so far is water. So I need to add, if you're following along on that little handout thing, I did 3.3 milliliters in my 0, then 3.2, 3.1, 3.0, 2.9, and 2.8 mils of water in each of these standards. So now what I'm going to do is I'm going to add my other reagents. I'm going to add my ascorbic acid. My ascorbic acid, I need 500 milliliters. So here's my ascorbic acid, and I am going to add 500 milliliters. And what I'll do for y'all is I will add the manufacturer's information for each of the reagents on Moodle so that you have them for your lab report. So that's my ascorbic acid. All right, and then after my ascorbic acid, I need ammonium molybdate. So my ammonium molybdate, and I need 0.5 mils of that one as well. So I just like to take them out and move them. Do you see how that one turned kind of yellow, right? This is why it's really important to have a 0 so you can see that difference. Okay. There we go. Now, I also need to add my perchloric acid. This is what you added to your samples when you were digesting the phosphate heads off, right? 0.65 milliliters. Okay, so I have two. I guess my hand's kind of in the way. Not done this before. Maybe I should have put it, I guess since I'm a lefty, I should have put the computer on the other side. Okay, so I have my, all of my reagents. Now what I need to put in is I need to put in my standard. So this is a known concentration of sodium phosphate. This is my 1 microgram of phosphate per 100 microliters of solution. And so what I'm going to do for this one is I'm going to add known concentrations so that I can create my standard curve. So the first one I'm going to do is my maximum, because that one is the 5 micrograms of phosphate, which is 500 microliters of solution. So I'm going to go ahead and add that one. 400. 300. 200. 100. And the last one gets none. Okay. All right, so now I kind of want to mix them up. I don't know if you can see the vortex there. It's hard for me to see what y'all can see, right? Give it a good mix there. Then we need to prep our samples. Okay, so those are our standards. We're going to incubate those in a second. But now we need to work on our samples. And I have stolen samples from a group. I'm not telling what group. Okay. Let's see. All right. So we can take all of these marbles off. Put them over here. Okay. So remember, we have our TLC samples in here, so everything you scraped off, and then you also already put perchloric acid in these. And so what you need to do is you need to add 3.3 milliliters of water to all of them, add 0.5 mils of ascorbic acid to all of them, and then 0.5 mils of ammonium molybdate to each of them. So that's what we're going to go ahead and do. And I think I have these in order. Yeah, 7, 6, 5, 4, 3, 2, 1. Okay, so this time we're putting 3.3. So 1, 2, 3, 3.3. Okay. Just right. Oh. Ooh. 3.3 of water. Okay, so we have water in all of them. So now what we need is our ascorbic acid and our ammonium molybdate. So we're going to go ahead and add that. So we need 500 microliters of each. And we're going to add our last reagent. I know, like, this is super exciting, right? Sorry you can't be here. I wish you could. All right, there we go. Okay, and then we need 500 microliters of our ammonium molybdate. Okay. And then we're going to give them a good mixing. All right. So we had our samples, we put our water, we put ascorbic acid, we put ammonium molybdate. So now what we need to do is we need to mix them, and then, I guess I need to turn this down, and then we are going to go and check the absorbance. Oh, and some of these you can already see. Can you see that? You see the difference? This one, so you can see there's another one that's got a nice difference already. Okay. Now what we are going to do, I'm going to take them over here. I'm going to take you with me. Let's see. And we're going to put them over here inside of these beakers that are sitting on the hot plate. So what we want to do is Put them in very carefully. Samples in one, and then we'll put our standards in the other. So we need to let these go about 5 minutes. So here are our standards. You can see our standards are starting to change a little bit too. I'm going to tilt them. Okay. All right, 5 minutes. And like I really need to do one of those, you know, it's like the cooking show and they like snap for you and it's all of a sudden done. That's what I'm going to do for you. Hang on. Okay, so there we go. We are all cooked. Can you see them there? So I'm going to turn this off. And what I'm going to do is you'll be able to see the color. Let's see. So that's our 0. Here's 1, 2, 3, 4, and 5. So what you should see if I can see this, is a nice gradient. I don't know if you can tell, something white in the background, but you can see a gradient of color. I have to find a white sheet of paper. So let me take these out. Oh, and look, here you can see if I can find a white sheet. Here we go. Here you can see that there's a gradient of color. So what we need to do is we need to vortex that again. So that's what we're gonna do. So we're gonna take all these samples out and we're gonna vortex them. Okay, so let's go and vortex them. Let's see if I hold this up for you with the white sheet behind it. Can you see now that there's a gradient of color? It's kind of yellow, 0 all the way over here to blue with the most standard in it. Okay, so I'm gonna take y'all over. Okay, so what we wanna do is we just wanna make sure that these are all the way mixed, especially while they're still warm. So just give them a good little mix. And then, now we want to put them in a water bath. So if you see over here, I have my bucket of water and I'm gonna leave them inside of the rack. And I'm just gonna go ahead and put that rack in that water to help them cool just a little bit faster. So now, 5 minutes. All right, so they look pretty cool. So what I'm gonna do is I'm gonna put my samples into cuvettes. So I'm gonna start with my standards. I wanna make sure I put the right one in the right container. So hang on. Okay. So my 0. Now remember, you only have to fill these cuvettes up. You see where I'm like, where's the camera? Right there, till about right there. That's what you need. So you don't really have to use a transfer pipette. You can just pour approximately what you need. And if you go over, it's okay. No big deal. Okay. Then 1. 2. 3. They look very nice in the cuvette. It's pretty easy to see because the cuvette box is white. I'll show y'all in a second. How awesome the standards look. There we go. Okay, so let's see if I can show you. You see there the range in color? So 0 is kind of yellow, and then all the way over here is more blue. So now I'm going to do my samples, well, your samples. I want to go in the right order, so I'm numbering them. 1 is LPC, 2 is SPH, 3 is PC, 4 is PSPI, 5 is PE, 6 is PA, and 7, they don't have a label, but you should have yours already done. Okay, so the first one is LPC. Okay. 2. 3. 4. Ooh, see, 4 didn't have very much in it. You can kind of see the color. Ooh, put it back too far. See how 4 is very light? Let's see, 5. We need 6. And finally, 7. Okay, so let's go and take this to the spec and see what we get. We have our standards, right? We have our 0, 1 through 5. We're putting it in, and we're going to read at 800. So start. Oh, we got a blank because our blank actually has a reading, so we're going to blank. And then we're going to start, and so we get a reading of 0 for our blank. And we're going to 1, standard 1, 0.2051. For standard 2, 0.4142. Oh, why did I do that? I read the same one twice. Dang. The standard 3 is 0.6194. Then 0.8096 for standard 5. And standard 6, 0.9970. Okay. Now we're going to put in our samples. Let's see if you turn this. I don't know if you can see. I'm going to swap out my 0. Make sure I'm going in the right direction. Okay, I'm going to make sure that we're pointed, our arrows pointing in the right direction. Okay. And we only get 1 through 6 in this cuvette holder. We'll have to do the last sample after we read these. Okay. So our first lipid sample, 0.0600. Lipid number 2, 0.2372. Lipid number 3, 1.2099. Lipid number 4, 0.2057. Lipid number 5, 1.0241. And then lipid number 6, 0.0460. Then I'm going to just swap out number 7 for number 6. Turn it that way. And number 7 is 0.1190. Okay, that's all your data.